And what we're showing here is about 17 days along, a little streak or groove starts to form in the top of the ectoderm, and a few cells from the ectoderm invaginate into that region and penetrate down into the middle and start forming, differentiating into the mesoderm. This only happens in higher organisms. Okay? Embryologists just love the word invaginate. Okay? They use it any chance they get. And so we see these cells invaginating into the middle area here. Okay? Surrounding the whole thing way out of sight is the chorion. Okay? And you can't ever forget that the chorion is out here. It will play a very important role later on because it becomes the fetal placenta. Okay? So there's really quite a bit going on in that picture. Okay, so the four fetal membranes are the amnion derives from the ectoderm, and I have a quiz question that somehow paraphrases every one of these. The yolk sac derives from the endoderm. The hindgut is kind of like the tail or derriere region of the, we call it embryo for the first 3 months and fetus for the second period of time, last period of time. And I used to be a French major, and I would call that a lantois, but I imagine a lot of people call it allantois. Just like I have a big problem every time I drive through Langlois, which everyone else calls Langlois. So anyway, chorion derives from the cytotrophoblast and completely surrounds the embryo and does all sorts of important things. It forms these little finger-like projections called chorionic villi that are the functional unit of the placenta. So amnion derives from the ectoderm. And we've talked about how if you imagine the ectoderm being on the top and the endoderm being on the bottom, remember our basic body plan is a tube within a tube. And so what happens, and of course you've got the amnion attached to the ectoderm, and as the whole thing folds around like that to form a tube within a tube, the amnion is pulled around the entire organism, isn't it? Because it attaches right here, and so as we fold it around, the amnion's pulled around the entire organism, and so totally surrounds and engulfs. Everything is inside it except for the chorion. Okay, and the yolk sac, on the other hand, is sucked up and eventually disappears, sucked up into the endoderm. The endoderm becomes the inner tube, which in a primitive organism we would just call it the gut, but in more sophisticated organisms evolves into a complete and elaborate digestive system. Okay. So there's the amnion. Okay. Nice pictures, the amnion. Okay, the yolk sac derives from the endoderm. As the endoderm folds around to form the gut, the yolk sac is pulled up inside the embryo and eventually disappears. Some blood cells and reproductive cells are formed in the yolk sac, but it's not very important. It's more important in animals that lay eggs, like reptiles and birds. Okay, the allantois or allantois basically evolves into the umbilical cord. Okay, the umbilical cord attaches to this hindgut region, which is sort of the embryological stalk that suspends the embryo inside the amnion, and that eventually becomes the umbilical cord. And we'll look at it, the umbilicus and umbilical cord in great detail. And become the umbilical vessel. The chorion derives from the cytotrophoblast, and this membrane surrounds the entire embryo and differentiates into the chorionic villi, which are the functional unit of the placenta and comprise the fetal placenta. Later in pregnancy, the amnion, as the embryo, at first, the chorion's way out here and the embryo's way inside. Okay, but as the embryo becomes larger, pretty soon the amnion becomes closer and closer and closer to the chorion, and finally, the amnion presses up against the chorion, and they sort of become one. Okay, and they become, and the whole works presses up against the inside of the uterus. Okay, and starts to distend the uterus. Okay, at about 12 weeks, the embryo starts to distend the uterus a little bit, press against the sides, and the uterus starts to change shape. Okay, they don't do this anymore, but I just show you this to show the amnion pressing against the chorion, which is pressing against the wall of the uterus. So this is fairly well along in pregnancy. And over here we have the placenta, which is very well developed at this point. This could be 30 weeks, 25 to 30 weeks. So, getting back to 17 days and looking at the top of the embryonic disc, and we've cut the amnion away so we can see down onto the top. You can see that primitive streak, remember that? And we're going to slice right through here and turn it 90 degrees so we can look at an axial slice of it. And this would be the embryonic disc, and the top is the ectoderm. Okay, and we're really only showing the ectoderm here. Here we're showing the whole thing. Okay, the ectoderm with the amnion attached, and the mesoderm in the middle, and the endoderm on the bottom with the yolk sac attached. This central part of the ectoderm is going to go through changes which ultimately will convert it to the central nervous system. So the central nervous system is derived from the ectoderm in a way, and all of these cells are going to differentiate into nervous tissue. Okay, and what's going to happen is the edges of this neural groove that you can see there, the edges of the neural groove start to form little shoulders. And pretty soon those shoulders are going to come together at the center, and you're going to wind up with a hollow cord-like structure which will give rise to the spinal cord. Okay. And once again, we've cut the amnion. This is only 2 days later. Okay. Once again, we've cut the amnion so that we can see down onto the surface of this and look at the changes that have taken place in just 2 days. And that's just 2 days there. So, okay, and this is 20 days. 1 day later, the edges have pushed over even more, and you can see how those edges are pretty soon going to meet, and that is going to close and form the spinal cord. And you can see this neural groove in the center here only 1 day later. You can practically watch this grow. It's a very small structure, but not so small you couldn't see it. And with some kind of magnification, you could actually practically watch these things happen in front of your eyes. 22 days, 2 days later, we've completely closed that neural structure, and it's going to give rise to the spinal cord and brain. Okay, and we have the surface of the ectoderm, which is going to become the epidermis, and these little structures have developed along the edge of the central nervous system. They are called somites. Okay, and we'll see what they're all about. But this neural tube is open at each end right now. This is going to become the head, and this is going to become the tail or hindgut. And you can see all these structures that are developing in the mesoderm region, and those so-called somites. Okay, so just a side view, and we know that we're going to fold around longitudinally, like I showed you, but we're also going to fold, then we're going to turn it sideways and fold it at each end to form a head and a tail. Okay. Craniocaudal folding. Here's just a side view, pretty schematic. Side view. Okay, and the way the folding that I demonstrated with the papers, and we have a tube within a tube there. And the endoderm is going to become the future digestive system. The ectoderm becomes the epidermis and the nervous system. And there you can see those somites and the craniocaudal folding, the head and the tail starting to take shape. Okay. Same sort of schematic with this neural tube, which will give rise to the brain and spinal cord, and the yolk sac is all the time becoming smaller. Okay, and so you can see how the ectoderm is being pulled around, and this is the amnion connected to the ectoderm. The amnion's way out here, but as the ectoderm is pulled around the entire structure like that, you can see how that amnion is just going to be pulled all the way around until it surrounds the entire structure. And the yolk sac's got to go somewhere, so it mostly goes up inside. Okay, and so now you see it's folded almost completely around. And we see the future digestive system, the yolk sac being pulled up, the amnion being pulled even further around, and the ectoderm is just about meeting on either side. And when it does, the amnion will have been pulled completely around. And there we see the ectoderm is pulled completely around. Are we showing the amnion here? No. Okay. The amnion's out here surrounding the entire structure now. When the ectoderm connected all the way around, the amnion, the edges of the amnion also connected, okay, but they are not a part of, physically a part of the embryo per se. They're not attached, and the whole structure, this amnion, is filled with amniotic fluid. Okay, but you can see how the amnion is pulled all the way around, and the yolk sac now has sort of disappeared, although it's still there, but it's not shown in this picture. This is an axial slice, and the yolk sac's not in the slice. And here we have the digestive system and the neural tube, so, and the epidermis on the outside, and the mesoderm the mesodermal structure shown in red, like the somites, consists of three parts: the dermatome, the myotome, and the sclerotome. And we'll see what they become. Okay, the sclerotome becomes the vertebra and ribs. The dermatome becomes the dermis, just underneath the epidermis. And the myotome becomes muscle. So the somites are derived from the mesoderm, and they differentiate into a lot of structures that are characteristic of higher organisms, such as mammals and animals like that. And for that matter, reptiles and birds. Okay, so somites. The endoderm at first is just a tube, but elaborates into a highly sophisticated digestive system. So even, this is probably at about, this is before 8 weeks. This is probably 6 weeks. Right here, you see a pretty sophisticated digestive system already. Oh, 5 weeks. Okay. So, in summary, the ectoderm differentiates into the epidermis and nervous system. The mesoderm differentiates into muscle, bone, and dermis. The endoderm differentiates into the organs of the gut, mostly digestive structures. And those somites, which are part of the mesoderm, become muscle and bone and that type of thing. So, the placenta, an organ derived from two individuals. The maternal placenta derives from the decidua basalis. The decidua basalis is that region of the inner layer of the uterus, the inner lining of the uterus, which resists the invasiveness of the embryo. Remember that? And and of course that becomes the maternal placenta. Okay, the decidua basalis is what exhibited this decidual reaction. Okay. And the fetal placenta derives from the chorion, more specifically the chorionic villi. Okay, and maternal blood pools in intervillous spaces. What does intervillous mean? It means between the fingers. So these little finger-like projections, the syncytial trophoblasts tapped into maternal blood vessels. So these chorionic villi are just, so we have maternal blood just running into tissue spaces, and the chorionic villi protrude up into maternal blood. The chorionic villi are a part of the fetal placenta. Blood never crosses the membrane though, okay, between the chorionic villus and the decidua basalis. There's a very thin membrane derived from simple squamous epithelium, the kind of tissue that we find in very small blood vessels like capillaries, only the little holes aren't very big, and there's no mixing of fetal and maternal blood. Okay. Certain, however, antibodies can traverse the placenta, especially IgG. Okay. So that's something to remember. So this is a great picture. Let's talk about this for a while. This structure here, what we're looking at there, is a single chorionic villus. Notice how we have the umbilical artery carrying, so the umbilical artery is blue in color because it's carrying junky blood from the fetus to the mom, okay? And except it's never going to arrive at the mom. It arrives at the chorionic villus and goes up into the chorionic villus and the vessels become capillaries, and waste products diffuse out through the walls of the capillaries into this pooled maternal blood, and so the waste products from the fetus diffuse into the blood and pass into the veins of the mother in the decidua basalis, the maternal placenta. In the meanwhile, maternal blood is flowing into the intervillous spaces, carrying fuel nutrients and oxygen, and the fuel nutrients and oxygen are absorbed into the capillaries and are carried back to the fetus via the umbilical vein. Okay? Remember, veins carry blood away from the heart, and so the umbilical vein carries good blood to the fetus, okay? And the umbilical artery carries junky blood from the fetus to the mother. And so this part right here, roughly, could be called the maternal placenta, the decidua basalis, okay, roughly, but not including the chorionic villus. And the chorionic villus strictly belongs to the fetus, okay? And this is maternal blood pooling in intervillus spaces here in the maternal part of the placenta. So this is happening all along this margin, the margin between the fetus and the mother. But you see, fetal and maternal blood are never mixed. Okay, so you can see here we have the umbilical stalk and about a 5-week embryo, the yolk sac, amnion, and you see the chorions way out here. And you can see chorionic villi and intervillus spaces. What do we find in intervillus spaces? Maternal blood. That's a good quiz question. Which of the following is found in intervillus spaces? Boy, if I don't have that in my test bank, I ought to.
It was just, yeah,
Just on our quiz.
Oh, it's on the last quiz?
Yep.
Oh, okay. Told you there was some overlap. Chorionic villus is the functional unit of the placenta. There you see a nice view of chorionic villus. This is about a 12-week embryo. Okay, and why would we be interested in doing chorionic villus sampling? Well, to count chromosomes, right? Okay, and we talked all about that, and we'll talk about it again. So, you know how the tech lines up the chromosomes of the homologous pairs, and you want there to only be 2 of each kind, and not 3 or some other number. And so, most commonly, they'll go in vaginally as the least invasive way to obtain tissue from chorionic villus. And so there you see a fully developed placenta with all the chorionic villi and intervillus spaces, and the decidua basalis out here comprises the maternal placenta, and of course that intervillus blood belongs to the mother, doesn't it? And the chorionic villi belong to the fetus and are part of the chorion. The yolk sac hasn't disappeared yet, but this is a very early, probably 5-week embryo. Another picture, early placenta. Okay. Probably about the same time. Okay, this is probably 8-week. Okay, and here we have 8 week, and at 8 weeks, there's still no distension of the uterus. Okay. But the fundus of the uterus is starting to be completely occupied by the embryo. Okay. 8 weeks. And here we have inches, so that you can see. This is a full-term placenta, very elaborate structure, derived from tissue from 2 different people. Okay, 35 weeks, fully developed placenta. So the decidua basalis, does that belong to the mother or the fetus?
Mother.
Mother. Okay. Nice MRI showing a vertex presentation. What's this right here? Cervix. Okay. And what do you think P stands for that we've been talking about? Placenta. And so, very important hormones of the placenta that you need to know about. One we've talked about already, human chorionic gonadotropin. Okay? Rescues the corpus luteum from degeneration until the 3rd or 4th month of pregnancy, at which time the placenta can take over and produce hormones. Okay? And the primary hormones that need to be produced are estrogen and progesterone. Okay? And this hormone maintains the endometrium, helps prepare the mammary glands for lactation, and things like that. One we haven't talked about, but a very interesting hormone. What's the strongest substance in the body?
Collagen.
That leather's made out of? Collagen. Okay, collagen is a protein polymer, a braided helix, and very strong stuff. And what the hormone relaxin does is it depolymerizes collagen. Of course, collagen is present in ligaments and tendons and in fibrocartilage, like the fibrocartilage in the symphysis pubis. And mothers may notice that they become very limber when they reach the later part of pregnancy. And it's because all of their ligaments and tendons are getting real stretchy because the relaxin is depolymerizing some of the collagen. The cervix is impregnated with collagen big time. The cervix is normally very tough and leathery and has no give to it at all, okay? And we don't want it to have any give to it because we certainly don't want the fetus slipping out of the uterus. And it's not about to do that because this cervix is not about to give way at all, okay? But during the latter part of pregnancy, under the influence of relaxin, all of that collagen, and we're talking really late in pregnancy, all of the collagen in the uterus is depolymerized, and the uterus changes its composition from being very tough and leathery to very rubbery and malleable, so that it can be dilated and effaced. Okay? Dilation and effacement are an important part. In fact, they're most of what goes on during labor. Okay? Dilation and effacement. And that can only happen when this tissue is very rubbery and malleable under the influence of relaxin. Babies also have very, their tendons are very stretchy, and of course they don't have any ossification around their joints either. And pretty hard, babies are, toddlers are falling all the time. And, it doesn't, most of the time, hopefully it doesn't hurt them because their joints haven't completely ossified. Then we have human chorionic somatomamotropin, which I just love saying. It helps prepare the mammary glands for lactation. It increases protein synthesis, which of course is required for a growing fetus. And it also exerts a glucose sparing effect on the mom with the idea in mind of saving the glucose for the fetus. Sometimes this happens a little too much and the mom will develop what is called gestational diabetes. And it's not an uncommon condition. I'm being asked all the time what causes gestational diabetes. And I very commonly say human chorionic somatomamotropin. So, okay. That's the reason. So, notice, this is the graph we looked at before. We were looking at it in connection with HCG, human chorionic gonadotropin. And this, of course, prevents the corpus luteum from degenerating. Notice also, since it's on this picture, we know that estrogen and progesterone levels at certain times in the menstrual cycle become very high. Okay, but at no time do they become so high as we see during the latter part of pregnancy. So this is a very profound elevation of both estrogen and progesterone during the latter part of pregnancy. And then, as if that weren't profound enough, then look what happens to estrogen and progesterone levels at birth. Bong. Basically all of the estrogen and progesterone was being produced by the placenta, and the placenta passes out of the body, and now we go from the highest levels ever experienced to essentially zero. So the effects of that have to be rather dramatic. Okay, and so the placental contribution to nourishment of the fetus are minimal to begin with because there isn't much of a placenta. But after about 3 months, we call this trophoblastic nutrition, and it derives from the word trophoblast, and it basically comes from the endometrial glands. Okay, and the endometrial glands give way